Hi, my name's Kert Viele. I am a statistical scientist at Berry Consultants. This is the January NHLBI ICTR webinar on enrichment designs. As an outline of what we'll be discussing today, we're going to start with a discussion of subgroups, some of the problems that come in when you're interested in subgroups within a population. We're going to focus on enrichment as an adaptive method for changing inclusion-exclusion requirements throughout a trial. So as the trial progresses, we're either going to be including more people or potentially excluding more people and gradually focusing on the subgroups that are the most promising. We're going to touch on final analysis methods and type 1 error control options, but we're really going to focus primarily on the enrichment as an enrollment mechanism. So we're going to spend a little bit of time on final analysis and type 1 error control, but that's really a topic that is broader than this webinar. And finally, we're going to show an illustrative example, which is the DAWN trial, which was published in the New England Journal of Medicine in 2017. So the whole purpose of why we consider enrichment is just uncertainty in the population. We usually would specify a trial population through inclusion-exclusion criteria. We want to include people which have a particular set of criteria, and then also we might exclude various people as well. And ideally, we walk into a trial with some idea on who we expect to benefit. So we can basically write these out and go, these are the people we expect to benefit. We are going to test this particular population. But there's often situations where you have uncertainty about that, where you can get all the investigators together and there's a discussion of, well, we think it benefits this set of people, but it might benefit this other group. Maybe some people that we would like to investigate, it doesn't benefit. And there's lots of uncertainty concerning, well, who should we actually include in the study? So some examples of this, if we're investigating how many hours since a stroke is a treatment still effective, usually in a stroke treatment, you talk about the time since a person was last seen well. And it may be that, oh, this treatment's going to benefit people as long as we can get it to them within 3 hours of the stroke, or within 6 hours, or within 12 hours. And there may be uncertainty about that. We're pretty sure that if we get it to them within 3 hours, that's what we want to investigate. But we have a lot of uncertainty as you get from 3 to 6 or 6 to 12, whether the patients are going to benefit. In oncology, we're often interested in a level of a biomarker that people need to benefit from a targeted therapy. If a therapy is aimed at a particular aspect of the cell, you might ask, well, how much needs to be expressed in order for the targeted therapy to be effective? Do we need a level of 3,000, a level of 5,000, a level of 10,000, whatever the scale is for that biomarker? And you could have a lot of uncertainty about that. We don't want to just pick a value and have that value be wrong. So you're really trying to manage two opposite mistakes here. So I've drawn on the bottom a kind of the broader oval is the entire population. And it's potentially divided into a group of people who benefit, they're in blue, and the outer group that remains in white of people who do not benefit. If I make the mistake of enrolling too narrowly, obviously I would like to just enroll the blue, but I don't know who they are in advance. If I make the mistake of enrolling too narrowly, and so I've got my enrollment in that little reddish rectangle, what's going to happen is I'm going to miss a lot of people who benefit from the treatment. I'd get this very narrow indication, and I wouldn't be doing as much good as I would like to. The other thing that can happen to you from a trial management standpoint is you get very slow enrollment. You may have this entire population coming to you, and you're going to screen fail everybody except for the people that are in that little red rectangle. So your trial may go a long time waiting for that narrow slice of people to come in. The other mistake that you can make is you enroll too broadly. So now this red rectangle is much bigger. It overlaps some of the people who benefit, but it also overlaps some of the people who do not. And so the concern here is that you're going to dilute your treatment effect. If you look at the average treatment effect across everybody you're enrolling, everybody in that red rectangle, you're going to get an average of whatever proportion of people in there who benefit, but then there's all the non-responders. And so because of that dilution of the treatment effect, you're going to end up with lower power in your trial. And in fact, even if you win this, if this were a phase 2 setting, for example, you might go on to phase 3 and you still have a lower power in phase 3. So this kind of mistake has carry-on effects as well. So we don't want to dilute, but we also don't want to enroll too narrowly as well. So the question is, well, how do we manage this when we have considerable uncertainty going into designing a trial? The basic idea of enrichment is you begin with a subset of the population. That subset, it could be a narrow slice. It could also be everyone. So you just need to decide this is where we're going to start. And you're going to conduct frequent interim analyses. And at each one of them, what you're going to do is you're going to look at however many subgroups you're interested in. You're going to examine which of those subgroups appear to benefit. And so everything, if you're looking at subgroups A, B, C, D, and E, you're essentially looking, does A benefit, does B benefit, does C benefit, does D, does E? If any of those are performing poorly, then what you're going to do is drop that poorly performing subgroup. So if A and B look like they're doing badly, C, D, and E look like they're doing okay, we might drop A and B and continue the others. It's also possible to add new subgroups at this particular time. So we talked about a stroke example. If our subgroups, we might build that up. We might start enrolling, say, 0 to 6 hours. And if things are looking promising in 0 to 6 hours, we might extend our window up to 12 hours. So you really can do this in both directions. I'm going to focus more on the start with everybody and drop, because that's more common, but it's not more correct. You can do it either direction. The goal of this, regardless of whether you're adding subgroups or dropping them, is to gradually focus on the subgroups where the therapy is effective. So at the end, you want to be enrolling in the places where the therapy is effective, and by doing so, get more patients in those groups. In terms of today, given this is a single webinar, we're going to illustrate these ideas for a simple biomarker positive negative setting. So I only have 2 subgroups, the positives and the negatives. And within that context, we're going to discuss the benefits and risks of doing this enrichment design. So focusing on this example, as a base case, I want to compare what a homogeneous population would look like. So if you didn't have to worry about subgroups, if you had a randomized sample of about 100 patients, so 50 in each group, 50 control, 50 treatment, that kind of trial has 84% power for an effect size of 0.6 while maintaining 2.5% type 1 error. As soon as we start to consider subgroups in this, everything is going to get more difficult. So I'm mentioning the homogeneous population, this 84% power, just as kind of a cap. As soon as I consider subgroups, I will not be able to get this 84% power. And anything that I do, what I'm really doing is I'm talking about, well, how close can I get back to that 84% power? That's kind of the maximum that anything can benefit here. In our example, Again, biomarker positive, negative subgroups. I'm going to consider that I have 70% of my population as biomarker positive, 30% is negative. So I've got all these little stick figures in here. You can think of my subjects that are arriving. These are all the enrollment over time. I've got a lot more blue, the 70% biomarker positive, and I've got the few reds, the 30% negatives that are also enrolling. Those would all be mixed and matched together. But as the trial enrolls, I get both of these populations coming into my trial. Anytime that I want to start asking questions about subgroups, does this work in positives? Does this work in negatives? I'm immediately asking more questions. So this is 2 questions as opposed to does this work in everybody? I'm also going to end up with a smaller sample size for each question. So my 100 subjects that I'd have when I consider a homogeneous population, as soon as I get subgroups, now I'm talking about a sample size of 70 for the positives and only a sample size of 30 for the negatives. So I'm asking 2 questions. My sample sizes are smaller. If you really want me to get everybody correct, I've now got to do 2 things instead of 1. All of these things come into your main summary. Your power is going down here, or you're going to need a bigger sample size and potentially a much bigger sample size. There is no magic bullet here that is going to let you investigate 2 subgroups at the same cost of investigating the whole population. You also now have to consider different possibilities for the truth. If you are analyzing a single trial in a homogeneous population, we usually worry about power. If the drug works, what's the chance that we get this correct? Or if the drug doesn't work, what's our type 1 error? As soon as I start talking about subgroups, I now have to consider more possible underlying truths. So that's what this particular table is giving you. It's possible that my new drug, my new therapy works for everyone. So I have this 0.6 effect size in both. Maybe it only works in the positives. So 0.6 here, no effect for negatives. Maybe it only works in the negatives. So flip that. 0.6 for negatives, 0 for positives. And it might work in no one. So 0 in each of these. So I need to start thinking about a trial that gets all of these situations correct. Ideally, whatever our underlying truth is, we're going to match that up. And of course, you could have some mix and match of these. So you could have an effect of 0.6 for positives, and maybe it works, but it doesn't work as well in negatives, 0.2. So you have some kind of off-target effects, for example. Okay. If I don't do any enrichment, so let me just slide back a couple slides. So this was my enrollment scheme. If I just go ahead and enroll all comers, whoever comes into my trial, I'm going to do an analysis at the end after everybody's involved. Well, what does that do for me? How successful am I? If the therapy works in everyone, and what I'm going to do is I'm going to separately analyze the large group and the small group. So I'm going to get about 70 patients in for the large group. I'm going to get a power of about 69.4%. Remember, it was 84 when I had a homogeneous population. So this is down. It's just reflecting the 100 going down to 70. The smaller group, it only has a sample size of 30. So its power goes down to 37.2%. This is all just reduction in sample size. So you can see anytime it works in positives, I get my 70% power. These situations where it works in the negatives is 37.2%, quite low. And I've got my type 1 error of 2.5% for these null cases where it doesn't work. So this is kind of a base. If I don't do anything with my enrollment, I'm going to suffer on power just from the smaller sample size. Okay, so the idea of enrichment is to counter this lower sample size in each subgroup. And we do that by, if we could stop some of the bad groups early, we could enroll more subjects in good groups. And so this is going to help us with differential effects. So our idea here is this was what our enrollment looks like when we have all comers. Suppose that our treatment only worked in negatives. It would be nice if we could go from this picture to something like this picture. And so that reflects that we did some kind of interim analysis right about at this point. And so to go back, we didn't enroll the rest of the positives. We concluded sometime early that the positives didn't work. And so we stop enrolling them and we fill up our sample with the negatives only. So from here on out, we just enroll negatives. I've drawn this so each of these two slides, so going back one and this, they both have 30 stick figures in them. But now we have a lot more red stick figures included, more negatives. Keep in mind that whenever you do this, this is deliberately longer. You can't change positive people into negative people. And so what's going to happen is you're going to have to enroll longer to enroll that smaller subgroup. So this is something you should always keep in mind. In addition to the statistical considerations, you have enrollment considerations. Once you focus on a narrow subgroup, it's going to take a while to enroll that subgroup. So, but the statistical advantage here is I'm going to end up with a lot more negative patients, these red patients, and potentially I can increase my power in that subgroup by doing so. The details of how this happens. So I'm going to begin this particular trial by enrolling everyone. I'm going to do periodic interims at 40, 60, and 80 subjects total. And at each of those interims, I'm going to allow myself to drop a subgroup if the probability of benefit is less than this threshold of 50%. So benefit just means that the treatment is doing better than the control within that subgroup. If both groups stop during the trial, if both groups get dropped, then the entire trial would just stop for futility and the trial will report this works for nobody. The timing of these interims and the threshold for dropping need extensive simulation. So the 40, 60, 80, I picked that based on some experience, but that might not be optimal. You might want to consider maybe only doing interims at 50 and 75, maybe 20, 40, 60, 80 instead of starting at 40. You should explore all of those kind of possibilities and essentially just pick one that is the best. So I don't know of any formulas that say this is the exact right time or threshold to use. We tend to simulate many possibilities and just try to optimize numerically. Okay, so at the end of the trial, you're going to end up with this enriched population. In the slide that I showed before, I ended up with those blue people at the beginning, and then I had a large number of the red subgroup as well. So ideally, this enrichment has produced more people in effective subgroups. You now have to decide how to analyze these. I could analyze the subgroups separately. That's what I talked about as the final analysis for the non-enriched population. I could pool all of the data. I could do something in between. All of these things are possible. I'm going to touch on these, but again, this is the part of the webinar. It's a little out of scope for what I want to discuss today. I'm really trying to focus on the advantage of enrichment to increase the sample size in effective groups. I'm going to touch on these possibilities at the end, but for the moment, I'm going to focus on how has this increase in sample size affected this analyzed subgroup separately analysis. And so the first thing I'm going to do is go, well, in theory, we're increasing the sample size. How much did we actually do it? You can see in this table, this is giving you the average in for each sample size for the large group and for the small group. The first number is the non-enriched design. So this is where we just enroll all comers and we analyze at the end. If we haven't done any enrichment, we expect all comers to average the population proportions. So 70% are 70 in the large group and 30 in the small group. And it doesn't matter what the underlying truth is. We're always enrolling all comers. As soon as I enrich, it makes a difference on what the underlying truth is. If, in fact, the therapy works for everyone, then what's going to happen here is we actually don't drop many groups. So we don't drop the positives very often. We don't drop the negatives very often. These results actually look very similar to the non-enriched. And the reason is it just doesn't drop anybody. So you can see the numbers are slightly smaller, 70 versus 69.6, 30 versus 29.5. Occasionally, we will make a mistake and spuriously drop things for futility, but not very often. And the times that we do drop groups tend to be situations they're not going to be successful anyway in this trial. In the null, where it works for no one, you can see the sample sizes are significantly reduced, 70 to 50, not so much in the small, the 30 to 28. It's hard to figure out what's going on in the small group because it's small. But you can see the sample size is reduced, and again, that's from the futility stopping. So we do recognize the large group is ineffective. The enrichment is aimed at differential effects. So you can see we get an increase in this positive group, 70 up to 76, and we get a decrease in the small group, 30 to 20. This works in the positives. So we do end up dropping the small group periodically, and by doing so, we can fill in the rest of our sample size with large group patients. If it works only in the negatives, so this was the little graphic that I showed, you can see the increase in sample size. So we drop the positives and do increase from an average of 30 to an average of 46.5, and we also drop out of the larger positive group. So certainly we do get the enrichment benefit is really this increase and also this increase. The subgroups where the therapy is effective, we get a larger sample size. That translates into power. So this, instead of being the average in, this is the power for each group. Again, works for everyone. We didn't make much changes. So comparing the non-enriched design to the enriched design, these are pretty similar, 69, 37, 38. These might be more simulation error than anything, but these are all close numbers. The type 1 error actually is reduced a bit, 2.5 down to 2.2 in both groups. And again, that's just from the futility stopping. That's helpful for type 1 error. We get our benefit from differential effects. So remember, the large group, it wasn't increased by that much. 70 to 76 was the sample size increase, and the power only goes up from 69 to 72. We did get a larger increase in the small group. So the power here is going from 37 up to 51, and that's reflecting our sample size increase from an average of 30 to an average of 46. So this is where the primary benefit is when you've got this, it works in a small group, you stop the larger group, get a big sample size in the small group, and get a power increase by doing that. Okay, so just to emphasize, why is this so much better? The smaller group, why is it so much better for the smaller group? So I'm trying to get at the notion of the big group didn't get helped that much, but the smaller group did. And again, this is just the smaller group is small. So any increase that you make is going to be proportionally greater. It's also easier to determine that the bigger group is bad and stop it early. It's really hard to get out of the small group. You can't figure it out that quickly. So you're going to see this benefit. This is a theme that's going to continue. If I wanted to recover the power, so recall I had 37% power for the non-enriched design when it only worked in the small group. If I end up with the enriched design, I got that up to 51. Let me go back. Yeah, so 37 up to 51, 52%. Suppose I actually wanted to get power numbers that got back up to the 84, what I actually started with a homogeneous population. That's going to require a much bigger sample size. So if you want to recover power for small groups, what you're going to need is, without enrichment, you're going to need 333 subjects to recover power. And that's just arithmetically aimed at, I need to get the sample size back up to 100 in the small group. So the small group is 30% of the total, 30% of 333 gets me back to 100. So that's a pretty big increase in sample size, 100 up to 333. Keep in mind, this is not a consequence of enrichment. This is a consequence of asking the subgroup question. If you want to know what's going on with 30% of your population, you're going to need to sample a much bigger group of people. The advantage of enrichment is it certainly does not get you back to 100, but you could actually use 240 patients with enrichment, and you will get back to that 84% power, even if it only works in the small group. So enrichment itself is getting you this sample size gain, 333 to 240. So this is a big adjustment here. It does not get you back down to 100. If you ask subgroup questions, you're going to need a much bigger sample size, but enrichment can have a meaningful impact on that. All right, so that was my main message, is that by doing the enrichment, you're focusing the enrollment over time. You can get more patients on the effective subgroups, and thereby you can investigate subgroups with a smaller sample size. I did that particular discussion for just analyzing the subgroups separately. There have been a number of alternatives that have been mentioned in the literature. You should certainly explore this. I can't get into the details of all of these, but I want to give a flavor of kind of what's been proposed and why you might want to do something a little bit different than that. So if you noticed, enrichment gained little for the works in both scenario. We weren't able to focus our enrollment. Usually we didn't drop any subgroups. And so that sample size loss for subgroups remains. One thing that people have proposed is, well, what if you pool the subgroups that are remaining at the end of the trial? And so what would happen if you think about our example just with biomarker positive and negative, what we're going to say is, if you get to the end of the trial and only one subgroup is enrolling, then you're just going to do the analysis on that one subgroup. So if you get to the end and only positives are left, then we analyze positives. It's no different than doing a separate. If only the negatives are left, we only analyze the negatives, no different from separate. But if you get to the end and both positives and negatives are enrolling, then we're going to pool the results. And so the goal of that is to try to increase power in this scenario where they work in both. And that does work. So the intuition does hold, again, not getting into all the details here. The disadvantage to doing this pooling of the remaining subgroups is you're going to get an increased chance of type 1 error when there's a differential effect. So if you can think of the scenario where it works in the large group, but it doesn't work in the small, it's quite possible that you will get to the end of the trial and you've never dropped anything. It was hard to figure out the small group was bad. And so what happens, you do your pooled analysis, you basically carry the small group along. And so you get a successful trial and you declare success in everyone, even though the trial or the therapy only works in the positive groups. So you can get this increased chance of type 1 error when you have differential scenario, particularly the works large scenario. So those are kind of the trade-offs. You get a gain in works both in terms of power, you get a problem for type 1 error. Another possibility that people have proposed is just to pool all of the patients regardless of who's enrolling at the end. So that may seem a little unintuitive. It certainly has the same kind of pros and cons as pooling the remaining subgroups. The reason for doing this is purely statistical. And that reason is it controls type 1 error in the joint null. So you can basically say I have a 2.5% chance of claiming it works for anyone when in fact it works for no one. So this notion of pooling all patients is really a statistical justification. In terms of all the medical aspects of this, clinical aspects, pooling the remaining subgroups probably makes more sense. But this has some advantages and has some it can sometimes smooth regulatory interactions, for example, because of that control of type 1 error. So this is a possibility as well. There's another possible analysis, which is based on hierarchical models. This is a Bayesian methodology that acts somewhere between separate analyses and pooling. And what it does is it borrows information between the positive and negative subgroups. And it does that in proportion to how much the subgroups agree. So it essentially says, okay, how well are the positives working? How well are the negatives working? If both of those seem to be working equivalently well or equivalently poorly, then what it's going to do is it's going to produce an analysis closer to pooling. And so that's going to help you in this works both scenario. If the hierarchical model sees differential effects, so it's working well in the small group, working poorly in the large group, then the hierarchical model will borrow less. This is a property usually referred to as dynamic borrowing. And so that's going to, what this is intended to do is limit some of that type 1 error problem that came from pooling. Generally speaking, hierarchical models will moderate the risks and benefits as opposed to pooling. You'll get less benefit in the works both, but you have less of this type 1 error inflation. And so that might create a better overall profile for a particular trial. So this is also a possibility. So, in that context, all of these are aimed at this, analyze the groups separately, pool the remaining groups, pool everybody, do the hierarchical models. These are all different possibilities, but they're all, I want to emphasize the distinction between how you analyze and the actual enrichment itself, which is how you control enrollment during the trial. So these are modular pieces. You can enrich and you can choose your final analysis separately. Another aspect that comes up when you're doing an enrichment trial is how to do type 1 error control. And I'll start by saying there is no consensus on type 1 error control. You can read FDA documents and it emphasizes you need to take care and handle this appropriately, but is not really prescriptive in how that's done. And so I think that's wise, given that the literature still seems to have a lot of controversy here. So I want to spend a few slides talking about what are the general issues involved in this. But again, not saying that one way is right, one way is wrong, because this really hasn't been settled in the literature yet. In a standard trial, when we talk about type 1 error, that is pretty well understood what we mean. Under the assumption that the drug works for no one, remember in a standard trial, we're assuming homogeneous effects, what we require is to have less than a 2.5% chance, this is one-sided, a 2.5% chance of saying the therapy works for everyone. So we make homogeneous decisions. We're assuming a homogeneous population. We're basically saying if the therapy works for no one, we want a 2.5% chance of saying the therapy works for everyone. So this is a 2.5% chance of missing the entire population. So we could apply this standard to an enrichment design. If the therapy works for everyone, what's the chance of missing the entire population? So that's a pretty lax standard because we're going to be talking about different subgroups. The flip side of that is, and you'll see this in lots of papers in the literature right now, is to do strict family-wise type 1 error control. And so what does that mean? That basically says you're going to restrict the chance of making any mistake. So you look at every single one of the subgroups and you calculate the probability that you say the therapy works in any subgroup where it doesn't. And we need the chance of making any of those mistakes less than 2.5%. This has a long history in multiplicity control. It is important in the context of avoiding people fishing for subgroups by testing many. So you wouldn't want somebody to go, well, my therapy, it's really just snake oil. And what I'm going to do is I'm going to test it in 15 different subgroups. Actually, I'm going to be smart. I'm going to test it in 40 different subgroups, and each one of them gets 2.5% type 1 error. I'll probably win one of them, and I'll get on the market and make a lot of money. So you don't want people doing that. So this kind of fishing, the strict family-wise type 1 error, says you can't do that. You can't make a mistake for anybody basically here. So that's valuable. This type of strict family-wise type 1 error, it has a disadvantage in terms of if you make that control at 2.5%. We said in a standard trial, we were protecting ourselves at 2.5%. If it works for no one, we get a 2.5% possibility of it working. If it works for no one, we're going to have control ourselves at 2.5% chance that it works for, to say it works for everyone. It's possible for us to make much smaller mistakes. So for example, what could happen here is, suppose I say that the therapy works in groups 1, 2, 3, 6, and 7. Lots of subgroups. And in fact, it only works in groups 1, 2, 3, and 7. So that fulfills this type 1 error criteria. We said it worked in group 6 and it didn't work in group 6. So we've made this type 1 error here. However, that's a much smaller mistake than what we originally were protecting ourselves against. The 2.5%, we were protecting ourselves. It truly works for no one. We say it works for everyone. This mistake, we missed group 6, but we got everything else right, is a much smaller error. So it's possible that we want to actually protect these kind of errors at a lower level. This is the kind of discussion that hasn't been fully, there's no consensus in the literature on how to do this. So that's what the issue is, and I'm not gonna get into more detail on that, except to say that if you want strict family-wise type 1 error control, that's a particularly expensive kind, and so you have to balance your resources against what you're trying to protect. Let me conclude here. This is the last section by giving a practical example, just to show that this has been done successfully. This is a trial called Dawn. It's going to be ironic because it has enrichment, but it did not enrich, but everything's been published. The Dawn trial tested thrombectomy and stroke after 6 hours. Thrombectomy had been shown to be highly effective before 6 hours, but it was unclear what happened after that window. If you're interested in seeing more details, the design paper is Joven et al. In 2017 in the International Journal of Stroke, and the results paper appeared in the 2017 New England Journal of Medicine. So both of these are published and available. The enrichment in Dawn is on the basis of infarct size. So Dawn, the clinical aspect, and apologies to all the clinicians where I'm going to butcher this right now, the basic idea of Dawn is that when you have a stroke, there's an area of the brain that is dead when the patient presents, but there's also an area of the brain that has lost blood flow, but is still alive, and you have the ability to save this. That kind of penumbra is sort of the mismatch there, how much brain that you can save. So that's what the Dawn trial was. They have a lot of inclusion criteria on that. The enrichment was on the size of the infarct itself. So the subgroups in Dawn, it allowed a prospect of infarcts from 0 to 50 cubic centimeters, and it had structured enrichment. So at any given time, the trial could enroll 0 to 30, 0 to 35, 0 to 40, 0 to 45, or 0 to 50. So you can think about this as subgroups that look like the figure at the bottom, where you've got 0 to 30, and then 30 to 35, 35 to 40, 40 to 45, 45 to 50. And what Dawn is really doing is it's allowing enrichment only from the top. So you can gradually remove groups from the top of this, and potentially you can add back. But you've got a sliding scale here. You can't drop 0 to 30. You can't enroll 35 to 40 unless you're enrolling everything smaller. So that's why I'm calling this structured enrichment. All of the dropping only happens from the top, and you could add it back if it made sense to do so. So structured enrichment. Interestingly, Dawn also used a different enrichment criteria than what I showed before. Instead of looking at the effectiveness in each of these subgroups, Dawn looked at something that was related. It looked at whether dropping the subgroups increased the predictive probability of trial success. So essentially, in deciding whether to go down, Dawn said, well, I could enroll 0 to 50 for the rest of this trial. If I dropped 45 to 50, and so I'm only enrolling the rest of the trial from 0 to 45, does that increase my chance of winning? And that's related to what we did before, because we're most likely to increase our chance of winning if 45 to 50 are performing poorly. But this is keeping it, it's a slightly different criteria for deciding whether or not to enrich. But again, the basic idea, if 45 to 50 is holding us back, it looks like it's performing poorly, we're going to drop it. And again, we drop from the top. So if we drop 45 to 50, we might also drop 40 to 45, and we could go down over time. There were a few other bells and whistles in Dawn. One thing that was interesting about Dawn is it actually never enriched, and it declared success at the earliest possible interim analysis. So essentially, Dawn claims success for everyone at the end. And remember, this is an important point. So, I mean, there's one aspect of this that I'm talking about enrichment, and I'm giving an example of a trial that did not actually use its enriching features. But one of the important aspects about enrichment, when you consider subgroups, you make it harder to necessarily be able to say it works for the population as a whole. And so I'm trying to emphasize this is an example where we didn't lose that. We were able to declare success for the population as a whole, even while we employed an enrichment design. So in summary here, the main point is that subgroups are hard. If you include subgroups, you're asking multiple questions. These questions involve small sample sizes. You're going to lose power by doing that. You're going to need to increase your sample size meaningfully to do subgroups well. Enrichment is an enrollment mechanism. This is intended to increase the proportion of patients in effective groups, if any are present. You can adjust the parameters of enrichment, how often you do interims, the threshold for enriching. You saw Dawn did something differently for predictive probabilities, but essentially enrichment does work for this purpose, especially in the situation where if you have small groups that are effective, enrichment will allow you to increase the sample size and actually detect those. Much easier than an equivalent non-enriched design. The final analysis in an enrichment trial is a user choice and something you ought to explore. We explored separate analyses, but I emphasized you could also pool under certain conditions or use a hierarchical model. And again, keep in mind that the type 1 error control, there are particularly lax standards, particularly strict standards. And as I said, the literature is still exploring if you're making a mistake that is a part of the population, how should that be weighed as opposed to making a mistake for all of the population? So you should certainly explore these and as necessary, consider regulatory requirements. Again, sometimes considerations are made in order to make sure that we get nice analytical type 1 error control, for example. So anyway, I hope that you enjoyed this and got some indication on how enrichment designs work and what are their pros and cons. Thank you very much.